Hello, this is KCIS New Taipei. Our project is called Delicious, and it aims to engineer butyrate-producing probiotics to combat vitamin D deficiency.
Vitamin D is involved in several health-regulating processes in the human body, including stabilizing immune function, protecting bones and muscles, and regulating the heart. Lack of sunlight exposure and vitamin D in daily diets are major causes of vitamin D deficiency. According to NIH, approximately 50% of the total global population suffers from vitamin D insufficiency today, indicating that the inactive form of vitamin D, 25-OHD, found in the body is below the optimal level, 20 nanogram per milliliter.
Current solutions to vitamin D deficiency include supplements and UV light therapy. However, they lack efficiency and effectiveness. For instance, UV light therapy is generally offered in an outpatient clinic, which requires patients to travel 2 to 3 times a week during business hour for treatment. This makes it relatively time-consuming and often inconvenient for the patient. Our solution to vitamin D deficiency utilizes the relationship between vitamin D receptor and butyrate. Vitamin D receptor, also known as VDR, is a nuclear ligand-dependent transcription factor that recognizes the consumed vitamin D, lets vitamin D bind to it, and regulate vitamin D-related genes. On the other hand, butyrate is a short-chain fatty acid synthesized by probiotics in the human gut. It increases VDR expression in a time and dose-dependent manner by upregulating the mRNA level through increased activity of the TGF-beta pathway.
To achieve the enhancement of butyrate-producing ability in probiotics, we plan to clone the bacteria by inserting our gene of interest, HBD and BCD genes, into our plasmids PET28A and PSB1C3. Transcription and translations of the BCD and HBD genes will result in the expression of butyryl-CoA dehydrogenase and beta-hydroxybutyryl-CoA dehydrogenase, therefore enhancing the rate-limiting step as well as another important step, and thus increasing the rate of butyrate's production. Increased amounts of butyrate upregulates the expression of VDR, increasing efficiency of vitamin D binding to the receptor and leading to the activation of VDR-regulated genes. Moreover, by adding the resulting bacteria to starters used to make fermented food included in the diet worldwide, we put our ideas into practice.
We flank the open reading frame encoded for BCD gene with a strong promoter, a strong ribosome binding site, RBS, and a double terminator. We also flank the open reading frame encoding for HBD gene with a T7 promoter, a strong ribosome binding site, and a T7 terminator. This construct design will lead to overexpression of butyryl-CoA dehydrogenase and beta-hydroxybutyryl-CoA dehydrogenase. The HBD gene was isolated, amplified through PCR, and inserted into the plasmid PET28A. The BCD gene construct was flanked with restriction sites and inserted into the plasmid PSB1C3. The engineered plasmid was then transformed into DH5 alpha. After using colony PCR to test the success of transformation, we extracted the plasmid and transformed it into BLRDE3 for protein expression. For the HBD gene, since lag operon is present, we added the IPTG to induce protein transcription. The induced culture is set as the experimental group, whereas the uninduced sample is treated as the negative control. The BCD gene, on the other hand, since lag operon is not present, we set engineered culture as the experimental group, while the wild-type culture as the negative control. We picked 3 colonies and ran SDS page with each colony grown under both induced and uninduced conditions. The SDS page result revealed that HBD, which has molecular mass 37 kilodalton, is indeed overexpressed in our engineered strain. Due to COVID-19, our team is opting in as a phase 1 team, and we only have 1 month in the lab. Therefore, we have only completed the experiments to this point. However, we plan future experiments that can further prove our concepts.
To ensure the visibility of our project, we plan to prove the HBD and BCD proteins' enhancement of butyrate synthesis by testing both rate-limiting steps. We will use NADH assay to test the protein activity and rate of reaction. More detailed explanation of the experiments can be referred to from our wiki page. After confirming the increase of butyrate, we further plan to test the relationship between butyrate and the upregulation of vitamin D receptor VDR expression. We plan to treat human colorectal carcinoma cell lines with varied butyrate dosage. HCT cell lines are used because the microbiome in the colorectal cells absorbs butyrate. If the result demonstrates that the VDR expression increases with the addition of butyrate in a time and dose dependent manner, it would confirm our hypothesis that our product, which enhances the synthesis of butyrate, could upregulate the VDR level and therefore increase the efficiency of vitamin D binding activity. And in modeling, we use models to explore the potential effects VDR upregulation can bring.
We built a ligand binding model to explore whether increasing the concentration of vitamin D receptor favors the binding activity between vitamin D receptor and its ligand, 125-OH2D. So when vitamin D receptor binds to vitamin D, they form a ligand receptor complex that later on binds with retinoids X receptor to form a heterodimer. This heterodimer then is capable of interacting with the vitamin D response element, recruiting proteins necessary to initiate the transcription of the target gene. We simulated dynamics using a system of OTEs. We extracted some of the parameter values, such as the degradation constant, from literature data. For the other unavailable parameters due to lack in accessibility, we substituted designated values for simulation. Since some of the parameter values are inaccurate, instead of building a model that accurately describes the time course evolution of each species inside our body, throughout the simulation, we will focus on comparing the change in the concentration of each species when a perturbation, or VDR upregulation, occurs. Our model shows that our fermented product is able to increase the transient state concentration of the ligand receptor complex, not the steady state concentration. This means that regular intake of our product is necessary. This finding aligns with the suggestions experts gave us during the interviews. However, this would not be a problem for our project, since our proposed implementation is to incorporate our engineered bacteria into fermented starter, which is what people eat on a regular basis. Our model also shows that our product can work with other vitamin D supplements synergistically. This opens another interesting application to our product. If a patient with severe vitamin D deficiency is able to use our product while taking vitamin D supplement treatment, our product will be able to generate a larger increase in the ligand receptor complex, making the treatment more effective, improving the health condition of the patient. Probiotic treatment has recently become a prominent area in the medical field. Building a theoretical model thus enabled us to explore the potential effect of our fermented product without actually conducting an experiment, which is unavailable for us due to COVID-19. Our model gives us insight on how our fermented product works and how to implement it. This is the end of our experimental part, and now we shall hand this into the human practice group.
We created a public opinion survey to collect the individual's knowledge about vitamin D and investigate the market. Before starting, we discussed with the experimental group to decide on questions that would help with the research and design the survey based on a logical order. After a few reviews, we sent our survey worldwide. The survey was open for 12 days in total from June 7th to the 19th. Once it ended, our members collected the data and analyzed the results.
Realizing the power of social media, we reached a wide range of audience from all over the world. Through Instagram, we were able to connect with other teams from around the world, such as Korea and Japan, to help promote our project as well as the importance of vitamins. We also utilize Instagram as a platform for education. Through our social media posts, we educated people about the importance of vitamins, including vitamin A and vitamin K, and the basics of synthetic biology. Our team collaborated with Team Korea HS, TS Taipei, and ASIJ Tokyo. We not only exchanged project ideas and progress, but also participated in TS Taipei's iGEM SDG Impact Challenge and Team Korea HS and ASIJ's Asia Collaboration Network. In the SDG Impact Challenge, we shared our SDG, which was 4.5, eliminate gender disparities in education and ensure equal access to all levels of education and vocational training for the vulnerables. In the Northeast Asia Collaboration Network, our team was able to connect with other teams around the world. We not only learned more about their project, but also shared our own project and received valuable feedback. Moreover, we exchanged educational pamphlets with ASIJ Tokyo, helping them translate into Chinese and sharing with our community.
To raise awareness for vitamin D deficiency, we want to teach people about this health issue. For example, we explain what vitamin D deficiency is, why it is important, and what we can do about it. We made 3 versions of our education presentation: an advanced version for people with a scientific background and adults, a simplified version for elementary to high school students, and a Chinese version for non-English speakers. We reached out to local communities and institutions to educate people on the importance of vitamin D, choosing specific communities and institutions with the goal of reaching a diverse audience. In addition, we post weekly education posts on our Instagram to introduce different kinds of vitamin to the public.
To help promote and successfully incorporate our product to the real world, we investigate the current market for potential customers and survey them to find the best fit way for the market. During the pandemic, people spend more time indoors because deficiency of vitamin D. Therefore, we create an e-commerce platform for customers to do online shopping. The product helps the user to get enough vitamin D while staying indoors and preserving user habits. More information was included in our marketing plan.
After researching some of the works from Dr. Michael F. Hollick, Dr. Edward Huang, and Dr. Joseph L. Napoli, we arranged interviews with them for better clarification on their research and seek suggestions for improving our project. These individuals' interviews allowed us to not only deepen our understanding about vitamin D, VDR, and probiotics, but also open us to more possibilities for our project.
Our wiki team is an essential part of our project. We spend a lot of time designing and implementing our website from the purpose of organizing all information into one comprehensive project. To start designing our wiki, we came up with a style guide that includes the color palette, font, as well as the font size for each heading. This is to keep the overall appearance of our wiki neat and organized. Then, we start our Figma web design by implementing the text, the graphs, and the figures. Although we built our wiki entirely from scratch, we gathered inspiration from previous iGEM wikis to help design and build our own.
As many others did, we used the Live Server extension on Visual Studio Code to help preview the appearance of our wiki page. After finishing designing using Figma, we implemented our website via HTML, CSS, and JavaScript, which are the basic building blocks for a static website. After putting up polls and some discussions with other teammates, we tweaked our designs to better match everyone's taste.
And above is the description of our project. For more information, please go visit our wiki page at 2021.igem.org/team KCS New Taipei. So, thanks for listening.
Great.